about looking at crop productivity. Stoller's only objective is to increase yields. We have no other objective. Increased yields is what we're all about. And so, Stoller is about 42 years in business. The first 25 years he worked very much with minerals, micronutrients, NPK, whatever, calcium. And about 15 years ago, he started spending a lot of time in the middle of the night reading scientific journals. And theoretically, he thought to himself, hey, just a moment, what is yield all about? And what drives yield? And what signals for good yield? And over the years, he learned that it really is plant hormones. The same things that drive us, our insulin, our thyroid, and all the other plethora of hormones that we use. We're biological, and so are plants. And so for 15 years, what we've been doing is studying how, especially exogenously, that means we can put something on the plant, can we increase yields beyond what we've been taught, what we've learned in the past. And I take one of your last slides where it's important to unlearn and relearn. And this is not easy because it's a whole new way of approaching things. I thought I was pretty good as a research scientist in Canada for 29 years until I came for an interview with Stoller. As soon as I came, in about 5 minutes, I felt about that view. I said, where have you been all your life? And what came through is that crop production, yields, are the result of signaling by hormones and small molecules. Wow. And is it easy? Well, 3 months after Stoller hired me, he came charging down the hallway, stood in front of me in my desk and said, Albert, when in the world are you going to learn this stuff? It's not easy. And therefore, if it takes a little while, I apologize, but that's exactly the way it is. And so it's a new way of thinking. I think it's the reality of crop production. In that, as soon as I came, I implemented what is called gene expression. And I apologize for that whole bunch of words that we have to spit out when talking about what we're doing. Gene expression is taking those genes that, for example, take up nutrients, those genes that move nutrients, those genes that utilize nutrients, and putting them all together and finding out what signals for the various aspects, and therefore getting the crop production that you think might be possible with your particular crop. Now, we have, what I was surprised at when I came with Stoller was he had about 300 different products. Now he has about 400. And I said, okay, I had just come from Harvard doing a sabbatical. And I was privileged to hear a particular PhD that had just finished his biochemistry degree, but was working with Venter. Venter is the person, private person that helped sequence the human genome at MIT. And he was working with Eric Landers, also part of the public input into sequencing the human genome. And he made what was called a DNA microanalysis. In other words, he looked at 25,000 genes at one time. And at the end of the session, he said, wow, guess how much this cost me? He said, $3.5 million. Well, I had the audacity, 2.5 years later when I joined Stoller, to ask the professor, how much would it cost me to do a DNA microanalysis? So I could look at all the genes, which ones are upregulated, which ones are downregulated, which ones stay the same. And the answer was $10,000. I said, wow, that's cheap. Now we do it for $500. So immediately we hired a molecular biologist, and he does nothing else day in and day out, and he's happy to do it. He looks at gene expression. So we have a very strong research backup. We have more field experiments, and that's how we progress. We don't sit still. We have new products coming on that'll just blow your mind. We have new products that we just tested very recently that do incredibly well when the soil is cold and the soil is wet. We have products that will increase the nitrogen efficiency of corn and soybeans, or corn in particular, from about 1 pound of nitrogen per bushel down to 0.66. Brand new data. We have products that will help increase the drought tolerance of plants. So these are the things that will be coming on into the future. What I'll be talking about now are those tried and true things that have been around for 10, 15, 20 years. Okay, so what we're going to talk about is essentially weather. And because weather gives you stress. And it's called abiotic, means that it's not a bacteria, it's not a fungus, it's not a virus. It's something that gives stress to the plant. And the only reason I was able to do this is the first time that I've ever done it. I tried to dissect, to separate the various effects of the weather or this stress, or whatever happens in the plant. Because we have the tools, we have the product, and because we have the molecular biology that has used gene expression to, gene expression to, I'm raising my hands instead of holding the mic where I should. Thanks a lot, sir, you're good. They use the gene expression to find out exactly what the product does. And therefore we have a tool. So in other words, it's like with a mechanic. You need a wrench in a particular size. You need a screwdriver and a particular type. You need a hammer and all of those. And we have so many of these quotation mark tools, and that's what we use. So for example, if we have wheat flowering, we know we can use cytokinin. And cytokinin stimulates cell division and cell differentiation. And it also stimulates development of the hormone auxin. And the auxin then regulates, from the scientific literature, we know, over 25% of the genes. So it's a very important hormone. So if you have wheat flowers, in other words, they're of different sizes and they're small and whatever else, then we know we have a tool for increasing the strength of the flower. So we go to the soybean plant, you look at the flowers, and wow, they're very variable in size. These look kind of limp. This is what we need. That's the tool that we need. If we have if we have an excess of ethylene, and ethylene buildup is one of the most damaging things that can happen. And ethylene buildup results a lot of time in diseases are off, but it also results in abiotic stress are off. And we can use the cobalt part of this cobalt to suppress the ethylene. And that's well documented in the literature and well documented by our gene expression molecular biologists. If we have an excess of energy particles, we need an antioxidant. And you often hear to keep your health good and to live a long and healthy and vibrant life, you need antioxidants. And you need to take them all the time. The same with the plant. It's a biological organism. And if you don't apply those antioxidants, you have a problem. Now, for example, when does this happen? Well, let's say conditions are really good, and you have the signaling hormone auxin says, great, we're ready, everything's in good shape, we're just going to go. We have sunshine, the right moisture and everything. And then all of a sudden, the clouds come, and maybe the rain comes. So all of a sudden, you have that huge amount of auxin saying, get fired up, and produce that energy, and you have that energy, and you don't have enough of the auxin because the weather changed instantly. So you have a lot of high energy particles. These high energy particles start destroying your mitochondria, which are the seat of energy production. They start destroying your cytoplasm, and as a result, you have a loss in potential productivity. And then what if we have, no, what did I do? What if we don't have enough sugar? In other words, here we look at, let's say the end of August, we look at all our pods, and we look at count the seed and say, wow, we're going to have 80 bushels for sure. Comes to harvest time, 45, 50. In other words, we haven't had enough sugar to fill those pods, to fill the seed. So we have to use a tool to do that. And then at the last, the micronutrients and whatever else. Okay, this is a complicated slide, but what it does is show how forgetting about germination and all the way to flowering, and forgetting about from R4 to the beginning, just looking at the reproductive stage. What can we do to enhance yield? Well, so we addressed these various issues, and we went from 60 bushels to 192. And this was winter production of soybeans in southern Texas. The planting was sometime beginning of October and just before the 1st of February harvest. Okay, so let's and do this abiotic stress. What does abiotic stress mean? Mostly it's weather. In other words, too hot, too cold, too rapid change, too wet, too hot, whatever it may be, and whatever relates to that. And just one step at a time. We'll go, okay, we'll go from wheat flours to ethylene, to excess energy, to not enough sugar, to micronutrient deficiency. And the only reason we can do this is because we've done the gene expression studies to show, and we have the field data to show that in fact this is what's happening. Okay, so what happens if we enhance the strength of this flour? And this is absolutely incredible. I'm forgetting the light for the soils. This is absolutely incredible. Instead of seeing a limp flower, a limp, L-I-M-P, and a small flower, and sort of bent over petals, you see one that stands up straight, a very large flower, and very stiff and well-formed petals. And so we've done this, and we I'm not so good at doing these things. I'm more of a theorist. My apologies. So anyway, we've taken wheat flours, and what we've done is created more cells. So in other words, if you think of the plant as workers, instead of having 20 workers building all these flours, you have 10 or 20 workers. So the productivity is that much higher. And once we have more workers or more cells, then we need energy to feed them. And so in theories, in fact, just about 2 weeks ago, we showed that the cytokine actually not only makes new cells and differentiates them into tissue, but transfers the sugar to where the new cells are. Fascinating. Okay, so we've done the first part here, which is the flour. Let's go to the next one, which is the apple. Okay, in this particular case, where we planted at the beginning of October and harvested just at the beginning of February in southern Texas. In other words, a winter crop of soybeans, something that nobody does. Anyway, we did it. We found that there actually wasn't too much ethylene. So there wasn't a real problem with disease. There wasn't a problem where ethylene could build up. And so our approach to use this cobalt molecule, cobalt atom in Berkeley wasn't any problem. But what happens if we put on this wheat flour 3 times? Once at pre-flour, once at R1, and once at R4. And in fact, from now on, I'll be putting on stacked treatments. So the first one is control. Next one is the cytokine for the wheat flour. On top of that is the cobalt for the cobalt. On top of that will be the excess energy. On top of that, sugar and so on. So we had about 8 different treatments. And so in every case it shows if you address this particular stress, how much better is your yield going to be? Okay, so then we go to too many high energy particles destroy cells and ages tissue. And in the wintertime, you can have fairly good, reasonable conditions, and then maybe down in southern Texas you get a hot day. So all of a sudden you have an excess of energy, and you don't have the right hormone balance to look after this excess energy. Or it may be 5 minutes left. Holy cow, I better hurry up. Okay, so at any rate, you have this imbalance of energy particles that can destroy your cells, destroy your mitochondria, and you don't have the right hormone balance. Well, there's many ways of attacking it, but one way is to put on an antioxidant. Now, the antioxidant that we ingest often are pomegranates, are cranberries, blueberries, strawberries. A lot of fruits or vegetables have the antioxidants. So, and when I came, because of my background also in food nutrition, I had our product analyzed for antioxidant capacity. It's incredibly very good antioxidant, one of the best in this private lab. Okay, and then what about enough sugar to moving to the pods and therefore developing the seeds? Well, we had a little bit bump there, and that helped bring it up from 110 to 113 bushels. And then, what about micronutrients? And we use the harvest more urea mate, which is an adduct type of calcium urea, which enhances nutrient uptake and used better than just the micronutrient by itself. In addition to that, it has potassium and everything. So in the end, what we did is go from 60 bushels to 119 just by looking after the stresses rather than the plant. And if I think of the stresses you have here in Illinois during the hot summer, I think maybe we need to address that. The question is, are we willing to go and try all these things? And with stress, here's one thing I have to point out. A little bit of the product multiple times is much better than a lot of the product. In fact, a lot of the product is going to choke the plant. You have to just gently help it along. It's like a sick person, and you gently help them come back and develop that strength that they already have. Now, this is from 2009. Down in Texas A&M, I shouldn't have said that, University of Illinois, I spoke about, in southern Texas. The director of the research center didn't want to make public what we did, but I saw that the leaves were getting a bit ragged, and so I had the technician put on 10 pounds of micronutrient. This is what they look like. The control looked like 10 days after application, and this is what the leaves look like. This is the control, and this is what the treated will look. By the way, we had 166 bushel yield here in this particular case. And the director looks at me straight in the eyes and says, "Don't tell me that in 4 days you can change a date from this to this. And if you don't stop spreading any of that word, I'm going to cut you off." Okay, fine. That's no problem. We can go on. The reality is the reality. Okay, so I'm coming into the 5 minutes, okay? If all of the above abiotic stress yield issues were not addressed, resulting yield would have been at control levels of 60 bushels instead of close to doubling the yield. This doesn't say that in every case you'll get that. A lot of cases, maybe you'll put on, let's say one of the first things we put on, and maybe get no effect. Because there is another aspect of the stress of the abiotic factors that's reducing your yield. That is the reality. There's no other reality. And remember, this was only in this segment, not from germination to flowering, but in that pre-flowering to R4, and we didn't look after the others. We don't stand still at Stoller. We have two crazy scientists. One is Gary Stoller, the other one is me. And we're always thinking beyond. And as I mentioned earlier, we have products that will look after drought tolerance like you've never seen before. We have products that at the end of the season will start moving more of the nitrogen and more of the sugars.